Welcome to the Deep Dive, your shortcut to mastering complex, crucial, and frankly pretty amazing topics. Today we're taking a deep dive into the incredible, sort of hidden world of the neuronal microenvironment. We're drawing heavily from the foundational insights of medical physiology by Boron and Boulpaep. It's a cornerstone text. Our mission, as always, is to untangle this dense academic material, make it clear, engaging, and, genuinely useful for you. Whether you're a medical student cramming for exams or just intensely curious about how your brain works, think of this as your backstage pass to truly understanding how your brain, that unbelievably sensitive supercomputer, maintains perfect stability and peak performance, even when, like the rest of your body, is in constant flux.
It's an absolutely critical system because the neuronal microenvironment is, well, it's basically everything that directly surrounds an individual neuron. So that includes the extracellular fluid, we'll call it the ECF, the tiny capillaries delivering blood, the huge network of supporting glial cells, and even, the neighboring neurons themselves. What's really fascinating here, I think, is that the ECF isn't some isolated little pond. It's in constant, really intricate communication with brain capillaries, with those glial cells, and with the cerebrospinal fluid, or CSF.
Right, this constant chatter. And why does this intimate dance matter so much?
Well, because this environment provides a meticulously regulated space. It's absolutely vital for your central nervous system neurons to function correctly.
So even tiny shifts can throw things off.
Profoundly. Even subtle changes in this finely tuned setting can really influence how your nerve cells behave. It's like trying to perform a delicate surgery on a vibrating table versus a perfectly stable one. The brain needs stability.
Makes sense. It's setting the stage perfectly.
Precisely. And the brain achieves this incredible feat through three major protective mechanisms we should probably cover. First, there's the famous blood-brain barrier, the BBB. Act like a really selective bouncer, meticulously shielding the brain's ECF from potentially disruptive ups and downs in blood composition.
Okay, the bouncer. Got it. What's next?
Second, we have the cerebrospinal fluid, CSF. It's constantly being made by a specialized structure called the choroid plexus, and it directly influences the ECF's chemical makeup. And finally, the often underestimated heroes, I'd say, the glial cells. They actively condition and fine-tune the brain ECF, making sure everything is just right for the neurons.
Okay, BBB, CSF, and glial cells. Let's maybe start with the brain itself. It's an organ that's surprisingly fragile, isn't it? Both physically and metabolically.
Oh, absolutely. Despite weighing, a fair amount, 1,300 to 1,400 grams, roughly like your liver, it actually has the consistency of, well, thick pudding.
Thick pudding. Not exactly built tough. So how does it survive rattling around in our skulls?
Well, its primary physical protection comes from being encased in bone, obviously. But crucially, it's also floating within that cerebrospinal fluid we just mentioned. The CSF acts as a fantastic shock absorber, really cushions the brain from everyday bumps and movements.
Okay, so CSF handles the physical side. But you mentioned metabolic fragility.
Yes, that's equally critical. The brain demands an astonishing amount of energy, but stores very, very little fuel itself. It has only about 5% of the glycogen your liver can hold. So when ATP, the cell's energy currency, gets depleted, cellular damage happens incredibly fast.
Yeah, I always find the brain's energy demand staggering. It's only, 2% of our body weight? But it just guzzles glucose and oxygen.
It really does. It consumes something like 15% of rest and blood flow, 20% of oxygen, and maybe 50% of glucose.
Wow, what on earth is it doing that needs so much fuel?
It's a remarkable energy hog because its primary demand is maintaining those steep ion gradients across neuronal membranes, especially through the tireless work of the NAK pump. Those gradients are absolutely crucial for neuronal excitability. That's how neurons fire and communicate. Plus, neurons are constantly turning over their internal support structures, their actin cytoskeleton. That takes energy too.
And the clinical impact of that reliance is pretty stark, right?
It's incredibly stark. Interrupt the oxygen or glucose supply, maybe from a stroke or cardiac arrest, and consciousness is lost within about 10 seconds. And irreversible nerve cell injury. That can set in after only 5 minutes. It's an incredibly tiny window for intervention.
5 minutes. That really underscores the need for powerful protection. Which brings us right back to that cerebrospinal fluid, the CSF. You called it one of the major guardians. So what exactly is this stuff?
Okay, the CSF. It's basically a colorless, watery liquid. It fills the brain's internal chambers, which are called ventricles, and it also surrounds the brain and spinal cord in the space just beneath the main protective layers. You can really think of the choroid plexus, the structure that secretes CSF, as the brain's own specialized kidney.
the brain's kidney. How so?
Well, it meticulously stabilizes the CSF's composition, much like your actual kidneys stabilize blood plasma. It's constantly filtering and adjusting, keeping the environment just right. Cleaning house, basically.
And it's not just sitting there, right? It moves around.
Oh yeah, it's a dynamic system. The CSF starts its journey in 4 ventricles deep within the brain, 2 large lateral ventricles, then a third and a fourth below them. They're all connected by these narrow channels like canals. From the 4th ventricle, the CSF flows out through small openings into what's called the subarachnoid space. Think of this space like a sleeve, wrapping around the entire brain and spinal cord. It's nestled between 2 of the brain's 3 protective membranes.
The meninges.
The meninges. Pia, arachnoid, dura mater. Exactly. The delicate pia mater is closest to the brain, then the cobweb-like arachnoid mater, and the tough dura mater is the outer shell. The CSF fills that subarachnoid space between the arachnoid and the pia. In an adult, the total CSF volume is maybe 150 milliliters. Not a huge amount, actually. Most of it, about 120 ml, is in that subarachnoid space.
Okay, constantly circulating. And you mentioned it acts as a shock absorber. This fluid cushion is really that important.
It's absolutely vital. It's one of the body's most ingenious protective mechanisms, really. The CSF dramatically reduces the effective weight of the brain. So instead of its actual weight, maybe 1,400 grams, it feels like less than 50 grams when it's floating in CSF.
Less than 50 grams. How does that work?
It's buoyancy. There's a slight difference in specific gravity between the brain tissue and the CSF. This buoyancy greatly diminishes the risk of acceleration-deceleration injuries. It's like having a built-in helmet, slowing the brain's movement gradually if there's an impact, rather than letting it crash against the skull.
And you definitely feel it if that cushion isn't there. I've heard about those awful headaches after a lumbar puncture, a spinal tap.
Precisely. After a procedure like that, if CSF pressure drops temporarily, patients often get severe headaches, especially when they sit or stand up. It's because the brain isn't properly cushioned anymore. Gravity causes these small movements that strain pain-sensitive structures like blood vessels and the dura, a very vivid demonstration of how crucial that buoyancy is.
So if it's so important, how much is made? And where does it go? Doesn't it just build up?
Good question. Most CSF is produced by those choroid plexuses in the ventricles, and they churn out an impressive amount, about 500 milliliters per day.
500 milliliters a day. But there's only 150 milliliter total volume.
Exactly. That means your entire CSF volume turns over roughly 3 times every single day. It's constantly being refreshed. This constant production creates a slight pressure gradient, pushing the CSF flow from the ventricles out into that surrounding subarachnoid space, and from there it gets absorbed back into the venous blood, specifically into a large vein running along the top of the brain, the superior sagittal sinus.
How does it get into the blood?
Through these specialized structures called arachnoid granulations or arachnoid villi. Think of them like tiny, pressure-sensitive one-way valves. They let CSF flow out into the blood when CSF pressure is higher than venous pressure, but they prevent blood from flowing back into the CSF space. It's a really neat mechanism.
Clever. So the absorption adjusts based on pressure.
It does. CSF formation itself isn't very sensitive to intracranial pressure, but CSF absorption increases quite steeply when pressure gets too high, maybe above 70 millimeters of water. This helps stabilize the pressure inside your skull. If that absorption system gets blocked or impaired, CSF can build up,
Leading to hydrocephalus.
Exactly. Hydrocephalus, or water on the brain. This can cause various problems depending on the type, issues with memory, incontinence, gait disturbances, especially in normal pressure hydrocephalus, or more acute pressure symptoms in obstructive hydrocephalus. Thankfully, treatments often involve inserting a shunt, a tube, to drain the excess CSF elsewhere in the body, like the abdomen.
Okay, so production, circulation, absorption. What about the CSS actual composition? Is it just filtered blood plasma?
No. It's far more sophisticated than just a simple filtrate. Its composition is actually significantly different from plasma. For example, CSF has much lower concentrations of potassium and amino acids, and almost no protein compared to blood. What's absolutely crucial is that the choroid plexuses rigidly maintain the concentration of key ions, potassium, hydrogen ions, magnesium, calcium, sodium, chloride, even if your blood plasma levels fluctuate quite a bit.
And that tight control is essential because
Because these ions directly affect neuronal excitability. Changes in potassium, for instance, can make neurons more or less likely to fire, so the brain needs this ultra-stable ionic environment. Think of the choroid plexus epithelial cells as tiny, sophisticated factories. They have a whole team of molecular pumps and channels like the NAK pump, various exchangers and cotransporters, specific ion channels, even water channels called aquaporins. They all work together in a very orchestrated way to secrete fluid into the ventricle. It's driven by the net movement of salt, like sodium chloride and sodium bicarbonate, which then pulls water along with it osmotically. Similar to how your kidney tubules work, actually, but sort of in reverse.
So it's actively crafting the CSF, not just filtering it.
Precisely. It's an active secretion process. And they even actively absorb potassium out of the CSF, helping to keep its concentration really low, around 3.3 millimol compared to about 4.5 millimol in blood. This low potassium is vital for normal neuronal function.
Okay, so we have the CSF bathing the outside and filling the ventricles. Now let's move closer in to the brain extracellular fluid, the BECF. That's the fluid directly surrounding the neurons and glial cells, right, in those tiny gaps.
Exactly. Even though brain cells are packed incredibly tightly together, those gaps, or clefts, are only about 20 nanometers wide, which is minuscule. This BECF still accounts for a surprisingly significant portion of the brain's total volume, maybe around 20%.
And you mentioned this volume isn't fixed.
Right. It's dynamic. It can actually reversibly shrink a bit, maybe down to 17% or so during periods of intense neural activity. That's because water can shift temporarily into the active cells. Now, solutes can diffuse through this narrow winding space, but they follow a much more tortuous path than they would in, say, a beaker of water. This complexity reduces the effective diffusion rate by about 60%.
So things move slower in the brain's ECF.
A bit slower, yes. And if cells swell up, for example, during oxygen deprivation or anoxia, the BECF volume can shrink drastically, maybe by half or more, and that really slows down diffusion, making it harder for nutrients to get in and waste to get out.
And that cell swelling links to cerebral edema, right? Which can be incredibly dangerous.
Absolutely. Cerebral edema is essentially the net accumulation of excess water within the brain tissue, often coming from the blood. Things like excessively high levels of the neurotransmitter glutamate or high extracellular potassium can cause neurons and glial cells to swell up with water, contributing to this edema. And the big problem is the skull. It's a rigid, inelastic box.
No room to expand.
Exactly. So any significant increase in the volume of brain tissue, CSF, or blood inside the skull rapidly increases intracranial pressure. And that can compress blood vessels, damage brain tissue, and quickly become life-threatening. The body has some compensatory mechanisms, like the Cushing reflex, but often medical intervention is needed. Things like hyperventilation to constrict brain blood vessels and reduce blood volume, or giving IV mannitol, an osmotic agent, to draw water out of the brain tissue.
Okay, so beyond just being the space between, what's the crucial role of this BECF highway?
It's the primary route for essential substances to get to the cells: oxygen, glucose, amino acids. And it's the path for waste products to get away from the cells, things like carbon dioxide or the breakdown products of neurotransmitters. It also serves a key role in intercellular communication. Molecules released by brain cells, neurotransmitters acting outside synapses, growth factors, other signaling molecules can diffuse through the BECF to reach adjacent cells, including glial cells or receptors on neurons that aren't part of a direct synapse. It's particularly suited for, slower, more widespread tonic signaling.
So how closely linked are the CSF in the ventricles and subarachnoid space and this BECF right next to the cells? Do they mix?
Oh, yes. They're in constant free communication. There's no real barrier between them. CSF can freely exchange with the BECF across the delicate pia mater lining the brain surface and across the ependymal cells lining the ventricles. Unlike the tight junctions of the BBB, these ependymal cells are leaky, connected by gap junctions but not forming a tight seal.
So that's why their compositions are so similar.
Exactly. It explains why the chemical makeup of CSF and BECF is very close, especially for small ions like potassium. And this free exchange, combined with the CSF's high production rate and constant circulation, creates a really efficient sink or waste management system for the brain. Metabolic byproducts, maybe leftover neurotransmitters, things diffusing from the blood, they can enter the BECF, mix with the CSF, and then get flushed out as the CSF is absorbed back into the bloodstream. Some waste products are even actively transported out by the choroid plexus.
Okay, so it's a highly regulated but still dynamic environment.
Yeah.
But even with all this control, when neurons are firing like crazy, there must be temporary changes in the ion concentrations right around them, in the BECF.
Oh, definitely. The ionic currents that underlie action potentials and synaptic potentials inevitably cause transient localized changes in BECF ion concentrations. Even a single action potential can slightly lower the extracellular sodium concentration and slightly increase the extracellular potassium concentration right near the active membrane, maybe by about 0.75 millimolar each. And if you have repetitive firing, like during intense activity or unfortunately during a seizure, these perturbations become much larger.
Potassium changes are particularly important.
Yes, changes in extracellular potassium, K plus O, are often highlighted because proportionally they're larger than the changes in sodium. And even small changes in K plus O can significantly affect the neuronal membrane potential. Remember, the resting potential is heavily dependent on the potassium gradient. So increases in K plus O can make neurons more excitable, potentially contributing to things like seizures if not controlled. And this accumulation of potassium in the BECF can actually act as a signal to nearby glial cells.
Signaling to the support crew.
Exactly. It can depolarize nearby astrocytes, for example, signaling the level of neuronal activity and potentially influencing glial metabolism or function.
Which leads us nicely to that third major protective mechanism, the blood-brain barrier, the BBB. You called it the bouncer. It was first shown way back in 1885 by Paul Ehrlich, wasn't it?
Yeah.
What's its core purpose?
Right. Ehrlich noticed that dyes injected into the bloodstream stained most organs, but not the brain or spinal cord. The BBB is essentially this remarkable gatekeeper that strictly controls what gets from the blood into the brain tissue. And it's absolutely necessary because, frankly, blood is not a stable enough environment for neurons. Think about it. Your blood composition changes after meals, during exercise, if you're sick.
Right. Hormones fluctuate, ions change.
Exactly. Fluctuations in amino acids, some of which are neurotransmitters themselves, or changes in potassium, hydrogen ions, hormones, inflammatory molecules. If these were allowed free access to the brain's ECF, they could wreak havoc on normal neural activity. As the Boron and Boulpaep text nicely puts it, without the BBB, running a foot race might temporarily lower your IQ because of the metabolic changes in your blood.
Huh. Okay, point taken. So it's a very strict barrier. But I seem to remember there are a few specific spots where it's intentionally leaky.
Yes, that's correct. There are a few small, specialized brain areas, mostly located around the ventricles, hence they're called circumventricular organs, or CVOs. Examples include the area postrema in the brainstem, which detects toxins in the blood and can trigger vomiting, or parts of the hypothalamus, like the median eminence, and endocrine glands like the posterior pituitary and pineal gland. In these specific spots, the capillaries are leaky, similar to capillaries elsewhere in the body. This allows neurons in these regions to directly sense the composition of the blood.
So they're like little windows for the brain to monitor the blood.
Precisely. They form critical parts of neuroendocrine control systems. They allow the brain to monitor things like blood osmolality or hormone levels, or to detect inflammatory signals like cytokines to initiate a fever. They also allow the brain to release hormones directly into the bloodstream, like from the posterior pituitary. Importantly, though, even in these leaky CVOs, there are often specialized ependymal cells called tannocytes that form a barrier between the local BECF of the CVO and the main CSF in the ventricle, helping to protect the general CSF composition.
Fascinating. Okay, so apart from these special zones, what actually makes the BBB so tight elsewhere? What's its structure?
It's really down to the unique properties of the endothelial cells that form the walls of the brain capillaries. Unlike capillaries in, say, your muscles or kidneys, which have small gaps, clefts, or even pores, fenestrae, allowing things to pass between cells, brain capillary endothelial cells are fused together by continuous belts of tight junctions. These junctions effectively seal the space between the cells, preventing most water-soluble ions and molecules from just slipping through. This seal is so good it results in a very high electrical resistance across the capillary wall.
So passage between cells is blocked. What about through the cells?
That's restricted too. Brain endothelial cells show much less transcytosis. That's the process of moving substances across the cell in little vesicles or sacs compared to other endothelial cells. They also have a high number of mitochondria, reflecting the high energy needed for the active transport systems they possess. And they're supported by a thick basement membrane in cells called pericytes. And very importantly, the end feet of astrocytes, those glial cells we mentioned, wrap almost completely around the capillaries. These astrocytes play a crucial role in actually inducing the formation and maintenance of those tight junctions in the endothelial cells, and they also help facilitate transport across the barrier.
So it's a multi-component barrier. Given this structure, what kinds of things can get across easily and what things are blocked?
Okay, generally, small, uncharged, lipid-soluble molecules cross pretty readily. They can just diffuse across the lipid membranes of the endothelial cells. This includes gases like oxygen and carbon dioxide, which are essential, but also many drugs, things like ethanol, caffeine, nicotine, heroin, methadone. They get into the brain easily because they're lipid soluble. Water also crosses relatively easily, partly by diffusion and partly through specific water channels called aquaporins, mainly aquaporin 4, located on the astrocyte end feet.
And what has trouble getting across?
Most ions like potassium, magnesium, calcium have very restricted access. Their movement is tightly controlled by transporters. Large molecules like proteins are almost completely excluded, and protein-bound metabolites like bilirubin are also kept out. What's also interesting is that the BBB has an enzymatic component. The endothelial cells express enzymes like peptidases and monoamine oxidase that can actually degrade certain biologically active molecules as they try to cross. A classic example is dopamine. If you give dopamine orally for Parkinson's disease, it doesn't work because it gets broken down by monoamine oxidase at the BBB. But L-DOPA, which is a precursor to dopamine,
Ah, dopa can cross.
yes, because L-DOPA uses a specific transporter, the neutral amino acid transporter, to get across the BBB. Once inside the brain, it's then converted into dopamine by neurons. It neatly bypasses that enzymatic barrier.
So the endothelial cells aren't just a passive wall, they're actively transporting specific things in and maybe out too.
Absolutely. The BBB isn't just a physical barrier, it's also a highly selective transport system. There are specific carrier proteins, transporters from the SLC and ABC superfamilies, embedded in the endothelial cell membranes. These transporters mediate the movement of essential nutrients like glucose, using GLUT1 transporters, essential amino acids, vitamins, and nucleic acid precursors into the brain. And there are also transporters that actively pump waste products or potentially harmful substances out of the brain back into the blood. Things like the P-glycoprotein transporter are famous for pumping out certain drugs, which can be a challenge for treating brain diseases. Plus, these endothelial cells themselves contribute to regulating the local BECF environment. They secrete some fluids similar to CSF and have transporters that help control local potassium and pH levels right near the blood vessels. It's a really dynamic interface.
Truly a complex and active gatekeeper. Okay, let's finally turn to those glial cells you mentioned earlier as the unsung heroes. Astrocytes, oligodendrocytes, microglia. You said they make up about half the brain's volume and actually outnumber neurons.
That's right. For a long time, they were sort of dismissed as just glue, that's what glioma means, or passive support structures, mainly because they don't fire action potentials like neurons do. They were considered electrically silent, in a way.
But that view has completely changed.
Completely. We now recognize that glial cells are essential, active partners in pretty much every aspect of brain function development, signaling, metabolism, protection, repair, you name it. And unlike most neurons in the adult brain, glial cells, particularly astrocytes and microglia, can still divide and proliferate throughout life. This is often stimulated by injury or disease, making them critical players in the brain's response to damage.
Okay, let's start with astrocytes. They have that star-like shape, right? What are the key jobs?
Yes. Astrocytes have these incredibly elaborate processes extending out, like arms of a star. These processes make intimate contact with both blood vessels, their end feet, wrap around capillaries, as we discussed with the BBB, and with neurons, particularly at synapses. This puts them in a perfect position to monitor and modify the neuronal environment. One absolutely critical role is providing fuel for neurons. Astrocytes are unique in that they store virtually all of the brain's glycogen reserves.
The brain's emergency energy stash?
Kind of, yeah. They can break down this glycogen into lactate. And then they can shuttle this lactate over to nearby neurons, which can readily use lactate for aerobic metabolism to generate ATP. Astrocytes can also take up glucose directly from the blood capillaries, convert it into lactate, and then export that lactate to neurons. This is often called the astrocyte neuron lactate shuttle. So they provide this sort of intermediary energy supply, buffering neurons against fluctuations in direct glucose delivery from the blood, especially during times of high neuronal activity. Lactate actually yields almost as much ATP as glucose when fully metabolized.
Fascinating. They're like the neuron's personal caterer. What about that potassium regulation you mentioned earlier, potassium buffering?
Yes, that's another huge role. When neurons fire action potentials, potassium ions rush out of the neuron into the narrow extracellular space, the BECF. If this potassium accumulates too much, it can make neurons hyperexcitable. Astrocytes are very effective at removing this excess potassium from the BECF. They have a high resting membrane potential, more negative than neurons, largely because they have lots of potassium channels, especially inwardly rectifying K+ channels. Cure. This makes their membrane potential very sensitive to changes in extracellular potassium. So when K+O rises, astrocytes depolarize, and this triggers mechanisms to take up the extra K plus a B.
How do they take it up?
They have several tools. The NA-K pump, of course, which pumps K plus in and Na plus out. They also have a NAKCl cotransporter, NKCC1, that brings all three ions in. And specific potassium and chloride channels are involved too. They work hard to keep extracellular potassium below a sort of ceiling level, around 10-12 millimolar, even during intense activity like seizures.
And they can spread this potassium load around.
Yes, that's where their network structure comes in. Astrocytes are extensively connected to each other by gap junctions formed by proteins called connexins. This creates a large functional network, or syncytium. Ions and small molecules can pass freely from one astrocyte to another through these gap junctions. This allows for something called spatial buffering. Basically, potassium entering astrocytes in an area where K+O is high can travel through the syncytium to areas where K+O is normal and be released there, or released near blood vessels to be carried away. It effectively dissipates local potassium hotspots across a larger area.
Wow, very clever system. What about their involvement with neurotransmitters, especially glutamate, the main excitatory one?
Absolutely critical. Astrocytes play a central role in the metabolism and clearance of neurotransmitters, particularly glutamate. They are key players in the glutamate-glutamine cycle. When glutamate is released at a synapse, astrocytes rapidly take it up from the synaptic cleft using high-affinity glutamate transporters, GL-LAST and GLT1. This is essential for terminating the synaptic signal and for preventing glutamate from building up to toxic levels. Once inside the astrocyte, an enzyme called glutamine synthetase converts the glutamate into glutamine. Glutamine is then transported out of the astrocyte and back into the neuron. Neurons then convert the glutamine back into glutamate, or GABA, the main inhibitory transmitter, replenishing their supply. So astrocytes are essential for recycling neurotransmitters.
And if that cycle breaks down?
Synaptic transmission fails pretty quickly, and it's clinically really important too. Under conditions like ischemia or anoxia, lack of oxygen, the NAK pump fails due to lack of ATP. Ion gradients run down. This can cause glutamate transporters to actually run in reverse, pumping glutamate out of cells, including astrocytes. Plus, neurons release massive amounts of glutamate. This leads to excessively high extracellular glutamate levels, which overactivates glutamate receptors on neurons, letting in too much calcium and causing neuronal damage or death. This is called excitotoxicity, and astrocytes are central to preventing it under normal conditions.
So they're protectors against excitotoxicity. Do they also respond to neurotransmitters themselves?
They absolutely do. Astrocytes express a wide variety of receptors for neurotransmitters, both ionotropic, ligand-gated channels, and metabotropic, G-protein-coupled receptors. So neurons can directly signal to astrocytes, and astrocytes aren't just passive listeners. They can actively modulate neuronal activity. For example, neurotransmitter binding can trigger increases in intracellular calcium within the astrocyte. These calcium signals can sometimes propagate as waves through the astrocyte network via gap junctions, or trigger the release of signaling molecules from the astrocyte itself, things like ATP, glutamate, D-serine, which are sometimes called gliotransmitters. These can then influence nearby neurons, enhancing or depressing their activity. It's a complex bidirectional communication.
So much more than just glue. And they provide structural and survival support too.
Yes, they secrete various trophic factors like BDNF, brain-derived neurotrophic factor, and GDNF, glial-derived neurotrophic factor, which are vital signals for neuronal survival, growth, and function. They are absolutely essential for synapse formation, synaptogenesis during development, and likely for synapse maintenance and plasticity in adults. If you try to grow neurons in a dish without astrocytes, significantly fewer synapses form. And as we touched on before, their end feet wrapping around blood vessels are critical for modulating cerebral blood flow. Neuronal activity leads to signals like potassium release or neurotransmitters that trigger calcium increases in astrocyte end feet. This causes the release of vasoactive substances from the astrocytes, leading to rapid changes in the diameter of nearby arterioles, usually vasodilation, increasing local blood flow to match metabolic demand. This neuron-gliovascular coupling is the fundamental basis for fMRI signals, like bold MRI, that we use to map brain activity.
Incredible. Okay, let's move to the next type. Oligodendrocytes. Their main job is myelin, right? The insulation.
That's their primary and most famous function, yes. Oligodendrocytes in the central nervous system, CNS, and their counterparts, Schwann cells in the peripheral nervous system, PNS, are responsible for producing and maintaining the myelin sheath. Myelin is this fatty, insulating layer that wraps tightly around neuronal axons. Think of it as the electrical tape for the nervous system's wiring. There's a key difference. One oligodendrocyte in the CNS can extend processes to myelinate segments of multiple different axons, maybe 15 to 30 or even more. Whereas in the PNS, one Schwann cell wraps around and myelinates only a single segment of a single axon.
And this myelin is crucial for fast nerve conduction.
Absolutely. Myelination dramatically increases the speed of action potential conduction along the axon. It allows the action potential to jump from one gap in the myelin sheath to the next. These gaps are called the nodes of Ranvier. This jumping conduction is called saltatory conduction, and it's much, much faster than conduction along an unmyelinated axon. Oligodendrocytes don't just provide the insulation, they also play a role in organizing the axon membrane at the nodes, clustering the voltage-gated sodium channels that are necessary for the action potential to regenerate at each node.
And this relates to recovery after nerve injury.
It does, unfortunately. In the PNS, if an axon is severed, Schwann cells can help guide its regrowth, and functional recovery is possible. But in the CNS, severed axons generally do not regenerate effectively. Part of the reason is that CNS myelin, produced by oligodendrocytes, actually contains molecules that inhibit axon growth. While this might help stabilize connections in the mature CNS, it's a major barrier to recovery after injury, like spinal cord trauma or stroke. Oligodendrocytes also have other roles, by the way. They're involved in pH regulation, as they contain carbonic anhydrase, and also in iron metabolism in the brain. And they are quite vulnerable themselves, particularly to glutamate-induced injury during ischemia.
Okay, in the last glial type, microglia, the brain's immune cells.
Exactly. Microglia are fundamentally different from astrocytes and oligodendrocytes. They actually originate from myeloid progenitor cells, like macrophages in the rest of the body, and they invade the brain during development. They're the resident immune cells of the CNS. In the healthy brain, they exist in a resting state, constantly surveying their surroundings with fine processes. But they become rapidly activated in response to almost any kind of brain injury, infection, or inflammation.
What happens when they're activated?
They undergo dramatic changes. They proliferate, change shape, becoming more amoeboid, migrate to the site of injury, and become phagocytic, meaning they engulf and clear away dead cells, debris, or pathogens. They also release a whole host of signaling molecules, including inflammatory cytokines, chemokines, growth factors, and potentially toxic substances like reactive oxygen species, free radicals, and nitric oxide. So while they are crucial for defense and cleanup, their activation can sometimes contribute to secondary damage, depending on the context. Microglia are implicated in the pathology of nearly all brain diseases: Alzheimer's, Parkinson's, MS, stroke, usually not as the primary cause, but as critical reactive cells that shape the brain's response to the initial insult.
Do they interact with the peripheral immune system?
Yes, they are considered the most effective antigen-presenting cells, APCs, within the brain parenchyma. If pathogens or abnormal proteins are present, microglia can process them and present fragments, antigens, on their surface to T lymphocytes, a type of immune cell that might have entered the brain, for example, across a compromised BBB. This helps initiate or modulate an adaptive immune response within the CNS.
Wow. Okay, we've really covered a lot of ground there. We've navigated the incredible, complex interplay within this neuronal microenvironment today. We went from the protective CSF and the vigilant blood-brain barrier right down to the really multifaceted, essential roles of these glial cells, astrocytes, oligodendrocytes, microglia. You've seen how the brain meticulously controls its internal composition, how incredibly energy hungry it is, and how even quite subtle changes in that environment can have profound effects on neuronal function. And hopefully it's clear now that glia are far, far more than just simple support. They're active, dynamic partners.
Absolutely. And as you think about all this intricate dynamic balance, the constant regulation, the protection, the communication, maybe consider this. Given the brain's incredible sensitivity and all these systems designed to protect it, how might subtle, long-term environmental factors, things like chronic low-grade inflammation or perhaps persistent dietary imbalances or even exposure to low levels of toxins, how might these subtly yet significantly impact brain function over a lifetime? Could they contribute to the development or progression of neurological conditions by slowly disrupting this delicate microenvironment? What stands out to you most about how the brain goes about protecting itself from, well, itself in the outside world?
That's a really thought-provoking question to leave us with. Mastering these concepts, understanding this microenvironment, is truly a significant step in your journey through physiology. It's complex, no doubt about it. But remember, you're not just memorizing isolated facts here. You're building an understanding of the intricate symphony that makes the brain work the way it does. You are part of the deep dive family, and you are absolutely capable of mastering this material. Keep exploring, keep questioning, and keep learning.